Yeah, you can start.
Yeah.
Please. Come on, people. Yeah.
Hello, sir. ഞാൻ സാർ ഡിസ്പെൻസ് സ്റ്റാർട്ട് ചെയ്തോളൂ സാർ. അപ്പൊ ഞാൻ എന്റെ ഒരു കാര്യം പറഞ്ഞേക്കാം. ജസ്റ്റ്
please unmute.
Good evening to all, and especially our thanks for Atul Nasa sir, Jayan sir, and Dr. B.K. Srikumar sir, and our president Dr. Sadish Kumar, Abdul Nasser, and our student forum. Today our student forum is going to be in all position for hosting the webinar, sir. So they will do all the thing from the beginning to the just we are giving the mic to the student forum.
Okay.
Please. And our special honor to Sanjay Mohan, also the speaker of the day. Okay, sir. Over to student forum people. Okay.
Thank you, sir.
Please.
Sir, sir.
Yes, please start. We can hear you.
Good evening. Hope you had a wonderful day. Evenings are beautiful. It comes with so many feelings. Look at sunset and see the beautiful colors of light. Logic is very important in our every person's life. Today is Jan 14, celebrated as World Logic Day. We human beings are rational animals. Using logic wherever it is needed has ensured the survival of our species since ancient time. It has also led to various technological advancement and growth of humankind as a whole. There have been several advancements in medical field in diagnosis, treatment, and prevention of various diseases. Today, we all are here for a special purpose under the umbrella of IPGA and associated with IPGA. I am Sneha, final year B.Pharm, Director, Member, and Media and Publicity, Students for IPGA Kerala State Branch. With me, Kirti Mohandas, final year B.Pharm, Director, Innovation, Students for IPGA Kerala State Branch. Now, I would like to welcome all of you to the national webinar on the topic Quality Assurance of IVD Equipment. Let's start the section with Seeking Blessings with Almighty. So I kindly welcome for a prayer song by Ashwati MB, final year B.Pharm, Director, Pharmacy Education, Students Forum, IPGA Kerala State Branch.
കലകൾ കാമാലകൾ കൈകോട്ട് ചാച്ചാടി കളിയാടി നിൽക്കുന്നു നവരത്നവേദി അവിടെങ്ങും അധ്യാത്മദീപം കൊളുത്തി അഭിമാനം കൊള്ളുന്നു അംബികൽ സ്കൂൾ വിജയിക്കൂ വിശ്വൈക വിജ്ഞാനരാത്രി വിജയിക്കാൻ ഞങ്ങൾ തൻ വിദ്യാലയദാത്രി അവിടുത്തെ മണിനാദം കരുണാമൃതമന്ത്രം അവിടുത്തെ മണിസൗധം ഞങ്ങൾ തൻ ക്ഷേത്രം ഹൃദയത്തിൽ വിനയത്തിൻ പുളകത്തിൻ പൂക്കൾ പദതാരിലർപ്പിച്ചു വന്ദിപ്പൂ ഞങ്ങൾ അവിടുത്തെ കാരുണ്യം തേടുന്നിതാ കനിയും അറിവാകും നൈവേദ്യം നീ.
Thank you, Ashwati. Now, I welcome for a welcome speech. I would like to hand over the mic to Devadarun PR, third year B Pharmacy, General Secretary, Students Forum, IPGA Kerala State Branch. I would like to welcome you, Devadarun.
Thank you. Good evening to one and all. Is it audible? Is it audible? Am I audible?
Yes, audible. Yes, audible.
Yes,
audible.
yes.
Audible.
Good evening, one and all, and it's a great pleasure to welcome you all on behalf of IPGA Students Forum Kerala State Branch. Before I move on to my welcome address, I would like to say a few words on IPGA. For those people who are new to this event, IPGA is a national level association which is one of the oldest and reputed professional bodies, which is formed in 1973. For the last 3 decades, it has been working for the upliftment of professional pharmacy. So I thank everyone who gave us the opportunity for conducting this webinar. So today, IPGA along with IPGA are conducting this webinar on quality assurance of IVD equipments. So most of us weren't that much aware of IVD equipments until recently. During COVID pandemic period, we became much more aware of our RT-PCR kit and antigen kit, etc. So today, for the inaugural function, we are honored to have Dr. Adul Kumar Nasa, who is the national president of IPGA, and he was retired on November 2022 as the head of Controlling Authority Drugs Control Department, New Delhi. So, and for his meritorious and faithful service, he received the Best Drug Controller Award, and I wholeheartedly welcome you, sir. Next, for giving the informative and valid session on quality assurance of IVD equipments, I welcome Mr. Sanjay Mohan KR, who is the AVP Business Development Agape. And next, I welcome Dr. Satish Kumar CS, President IPGA Kerala State Branch, who was the former drug controller, and now he is currently working as the head of Kerala, head of Business Development Agape Diagnostic Limited, Cochin. Next, I would like to welcome Sri PM Jain, sir, drug controller, Kerala State. I welcome you, sir. Next, I welcome Dilip, sir, who is the General Secretary, IPGA, Government Drug Analyst, Drug Testing Lab, Thrissur. I wholeheartedly welcome you, sir. Next, I would like to welcome Dr. Srigumar PK, who is the President of KPGA, former drugs controller, and I welcome you, sir. Next, I welcome Dr. Mahalakshmi, IPGA Women's Forum, Kerala State Branch. I welcome you, ma'am. Next, I welcome Dr. Nishit MC, Joint Secretary, KPGA. I welcome you, sir. And I welcome Dr. Sunil Kumar, Treasurer, KPGA. And I welcome you, sir. And I warmly welcome all the office bearers and members of IPGA and KPGA who are conducting in this webinar, who are attending this webinar. And once again, thank you all for giving us this opportunity, and enjoy the event. Thank you.
Thank you, Devadarun. For presidential address, I would like to invite Anushraya M, Final Year B Pharmacy, President, Students Forum, IPGA Kerala State Branch. It's over to you, Anu.
Thank you, Sneha. Good evening, one and all. Our scholars, a group of young leaders devoted to carrying our pharmacy profession and association into the future. What a great juxtaposition, thinking about our coming years in the place our first years began. That was the point. Healthy organizations don't get stuck in the past or get lost in the moment. They look ahead. On behalf of IPGA in association with KPGA, today we are organizing the national webinar on the topic Quality Assurance of IVD Equipment. An IVD medical device is an in vitro diagnostic medical device intended to be used for the testing of human samples where the results can assist in clinical diagnosis or management. An IVD device could be a test kit, reagent, software, system, or instrument such as a qPCR cycler. During COVID pandemic only, we all were aware of RT-PCR. So, the importance of IVD reagents and IVD equipment are more relevant in the current scenario. The in vitro diagnostics market size was estimated to be US dollars around 900 million in 2019 and is expected to surpass 1.4 billion by 2030. IVDs are broadly employed for the detection and therapy of several medical illnesses. IVD quality controls are samples used to authenticate the trustworthiness of IVD testing system to guarantee the precision of test outcomes and estimate the effect of factors such as environmental conditions and operators' performance on test outcomes. The global IVD quality control market is segmented depending on application, type, end use, and geography. By application, the IVD quality control is divided into immunochemistry, hematology, clinical chemistry. The type segment is further divided into quality control, quality assurance services, data management, and based on end use, the market is segmented into hospitals, labs, home care, and others. So I hope the coming session will much more interesting as well as informative to everyone. Thank you, everyone.
Thank you, Anushya. Now, we are entering into a much awaited session. Let's have a glorious beginning with the inauguration ceremony. It's my privilege to invite respected Dr. Adul Kumar Nasa. Dr. Adul Kumar Nasa was retired on November 2022. He was the head of office Controlling Authority, Drugs Control Department, Delhi. He received the Best Drugs Control Officer Award from All India Drugs Control Officer Confederation in appreciation of meritorious service in the Drugs Control Administration. The award was given during the inaugural session of 69 Indian Pharmaceutical Congress held at Chikara University, Rajpura, Punjab. During the conference, he also received fellowship of Indian Pharmaceutical Association on 23/12/2017 for distinguished and dedicated service in the profession of pharmacy. He has more than 25 years of experience in the enforcement of Drugs and AMP Cosmetics Act 1940 and rules made there under Drugs Order and Drugs and AMP Magic Remedies Act. He has participated in various seminars, conferences organized by WHO, CDSCO, other industry associations on the various topics related to drugs regulatory affairs. He is a member of Drugs Consultative Committee. He has been nominated as a member of Pharmacy Council of India under Section 3H of Pharmacy Act by Government of NCT of Delhi in March 2017. Currently, again elected as our National President IPGA. With much respect, I welcome you, sir, for the inauguration ceremony.
I think our guest has just left. I think some connection issue with Nasaji's car. Just wait for a minute. Dilip, can you call him?
Sir, in the meantime, Jain sir can because he's the dress controller of Kerala.
Yeah.
And he can tell a few words.
Yeah, Jainji, please.
He was the General Secretary of IPGA. Just for the time being only, I am holding.
Yeah.
Sir is the General Secretary of IPGA.
Jain, please continue. Nasaji,
Okay, sir.
Then, sir, Nasaji will continue, okay?
Nasaji?
Okay.
Nasaji? Yeah. Yeah, you have been introduced properly, Nasaji. You can start now.
Some technical issue was there. I'm sorry.
Yeah. Now it's okay. You can start because you have been already introduced by the.
Okay. Thank you very much. I'm audible.
Yeah.
Yes, sir. Yes, sir.
So, sorry, it was some technical issue, but anyway, thank you very much. Again, I have joined. So, very good evening to each and everyone. And at the outset, my heartiest congratulations to the young team. That is the student form of IPGA Kerala branch. That they, in association with Kerala Pharmacy Graduate Association, has organized this webinar on quality assurance of IVD equipments. My heartiest congratulations to very young President, Miss Anushriya M. And along with the very dynamic General Secretary, Mr. Deva Tharun VR. Then we have got Miss Sneha C. Trayerar, Miss Kirti Mohandas, the Director Innovation, Mr. Bharat Narayan M. Joint Secretary, Miss Amrita S. Director Professional Relation, Ms. Shamal, Vice President, Student Farm, and all the office bearers of the IPGA Kerala branch, Dr. Satish, the Drug Controller of Kerala, Mr. Jain, Mr. Dilip, the General Secretary, Mr. Shri Kumar, Dr. Mahalakshmi, Dr. Nishit, and all other participants. My all the best wishes to the young dynamic team that they have taken this initiative on a topic by inviting a speaker, Mr. Sanjay Man KR, who is the Associate Vice President of Business Development of Agape. And he's going to talk on quality assurance of IVD equipments. During this, COVID time, we have seen that all these, COVID-19 testing kits, including other kits like HIV kits, blood glucose kits, and other, devices, they have supported a lot in healthcare system. In vitro diagnostic medical devices regulations are well in place. So by today's speakers, let us see how the quality assurance of this IVD equipment is there, because this is a very important part of of quality control system. So my special thanks to Mr. Sanjay Man KR, the Associate Vice President, who is going to deliver on the topic on quality assurance of IVD equipments. With these remarks, I welcome each and everyone, and my best wishes to young and dynamic team. My special request to the young and dynamic team of IPGA Student Farm of Kerala branch, keep it up these activities. This will morally boost all of you, and future is very bright. And in the coming time, this medical device industry or the other pharmaceutical industry has got a lot of scope for the opportunities for the young students. So my best wishes to all of you, and best wishes to all the participants. And I wish this webinar will be a great success. Thank you very much.
Thank you so much.
Thank you, sir.
Thank you
So in
so
the
much, sir, for
Okay.
thank you so much, sir, for inaugurating our function and for your kind words. Next, we move on to our scientific session. For welcoming our guest of honor and speaker, I call upon Sneha C, Final Year B Farm, Prime College of Pharmacy, Treasurer, Students Home, IPGA, Kerala State Branch.
Before going to Jain sir, please talk 2 words, sir.
Please.
Thank you, Mr. Dilip, Sadish sir, and Dr. Nasa sir. I take this opportunity to congratulate the office bearers of IPGA Kerala branch for organizing such a seminar in the in vitro diagnostic medical equipments, and also request all the participants, probably students, to take this opportunity to improve your knowledge about IVD diagnostics. I am not taking much of your time. You are eagerly waiting for the presentation from Dr. Sajimon. Thank you for giving me a chance for addressing the invitees. Thank you.
Please continue.
Thank you, sir.
Like to
Good evening to
welcome
one and all.
I would like to welcome Devadarun for a kind words.
Excuse me, everyone. I have a small apology to make. I missed out 2 persons. I missed out to welcome 2 persons. Mr. Abdul Nasser, who is the General Secretary of IPGA, and Dr. Nishit MC, who is the South Zone President of IPGA. Sorry, I missed out that 2 persons. Sorry.
And our coordinator also, Abdul Nasser. Okay.
I mentioned him, sir. Abdul, moderator Abdul Nasser, sir. Sorry, sir.
It is Nasser, you mean, isn't it? Same. Abdul Nasser.
Okay.
So it's okay, yeah.
Okay.
Thank you. Thank you, Devadarun.
Now I would like to call upon Sneha for addressing speak.
Good evening to one and all. It is pleasure is our speaker. Started Medila in 98 and started the career in. He diagnosed in the year 2000. Care of business. He attended major international conferences like Medica in Germany, AACC in US, CMEF and CACLB in China, MedLab and Arab Health in Dubai. He is the author of GLB section in Tech Agape, the technical magazine of Agape from the beginning. He had in India and abroad. In Mumbai, TBA 120 FR or 40 in Japan in the year 2000 time. ISO 15189 2000 Quality Council of India in the year 2000. CA 270 Clinical Chemistry Analyzer in Furono Electric Company in Japan in the year 2012. TMS 301 Clinical Chemistry Analyzer in Tokyo, Koike Medicines INC, Tokyo in Japan. Next, it's my pleasure to welcome you to the session, sir. Thank you.
Thank you, Sneha.
Over to you, Sanjay Manji.
Yeah, Sadish, thank you very much. And first of all, thank you for all the participants who joined this late evening to this scientific session. So I will just share my screen. Just a minute. Is the screen visible?
It's visible. You can make it in the side mode. Okay,
yeah.
fine.
Okay.
Yeah, it is visible,
So,
sir. It is visible.
So we will start the sessions, that is the quality assurance in the IVD instruments. This session is a continuation of the session, but we have performed maybe 6 months back in quality assurance in the IVD for the reagents. Now, today we are taking a small session, a small scientific session on the quality assurance, how we do in the IVD instruments. When we look at IVD, in vitro diagnostics, the IVD is nothing but the test that can detect disease, condition, and infections. And in vitro simply means outside the body, which means in a glass, meaning these tests are typically conducted in tubes and similar instruments outside the body. The importance of IVD testing is that they allow the doctors to diagnose patients effectively and work to provide appropriate treatment. And today, in today's world, more than 70% of the diagnosis is based on the IVD testing. So today, the scenario is like that. Each and every doctor, he depends on the diagnostic test to properly diagnose and to give the treatment. So there is a lot of importance in IVD testings today. Like in one of the earlier speakers said that about the market of in vitro diagnostics in India, and it is estimated on a $1.8 billion is the India medical tech industry, which is valued in India in the year 2020. So there is a huge market also is there for the in vitro diagnostics. And fortunately or unfortunately, we are depending upon other countries, especially for the in vitro diagnostic instruments. And there are few companies in India who is actually doing their way to make India Atmanirbhar in IVD segment. Now let us understand what is an IVD instruments. I have seen, I think you have seen some instruments over here. These are different instruments which is used for different purposes. Say on the left hand side, on the top, you can see a specific protein analyzer. On the left hand side, left, you can see a hematology segment. On the bottom, you can see the semi-auto biochemistry analyzer. On the other side, you can see a fully automated system. On the bottom line, you can see a coagulation analyzer, and another one is a simple, blood glucometer. So these are a few examples of IVD instruments which we have taken for today's discussion. It is not the entire product. We have taken only few examples for our better understanding. And when we look at the consumables, in vitro diagnostic consumables, there are multiple type of consumables available. One is the liquid chemistry reagents. We call it as wet chemistry reagents. Another is something called as we call it as a biosensor electrodes. You can see the blood glucometer, and this kind of instrument actually they use a biosensor electrodes. On the other side, you can see all-in-one cartridges, and on the top side, you can see an RDT or a rapid diagnostic test based on immunofluorescence using capillarity or lateral flow immunofluorescence. So there are multiple technologies involved in the IVD, whether it is instruments or reagents. We call it as collectively as consumables. Now, when we look at the instrument classifications, the IVD instruments are basically classified into 4 different categories. One is called as a wet chemistry instrument. Another is called as a dry chemistry instrument. What is a wet chemistry instrument? Means the instrument which is actually uses the reagents in liquid format like glucose, cholesterol. All those reagents are provided in the system in the liquid format. We call it as liquid stable reagents, or sometimes we call lyophilized reagents, and these lyophilized reagents needs to be diluted with the respective buffer solutions to make the working reagent, but still the reagent formulation is in liquid format. That is called as wet chemistry and wet chemistry instrument. And if you look at the world IVD population, more than 90% of the instruments available globally is into wet chemistry, whether it is Indian origin or foreign origin. And another is a dry chemistry. In dry chemistry, what we do is that there are certain slides, something like it is photosensitive slides, and when we put a drop of blood or a drop of serum or plasma into that slide, there will be some reaction which happens on the slide, and that reaction in turn is measured, and that gives you an idea about the analyte in that blood. So that is called as dry chemistry systems. The major difference between a wet chemistry and a dry chemistry is that in wet chemistry, all the reagents which is used for the testing is in liquid format, and in dry chemistry, it is in dry powder format. Now, the other classification is an open system and a closed system. What is an open system? Means a system which can use any brand of chemicals. Say, for example, most of the semi-automated analysis available in the market are open system, which means if I'm a laboratory, if I'm going to do some testing, I can buy X company instrument and I can use Y company reagents, or I can use Z company reagents, which means I'm not bound to any company for the consumables. That is called as an open system. And there are automated instrument, fully automated systems also available in the open chemistry, but 99% of the semi-automated system in the industry are open system. What we call it as an open system. Now, the second is a closed system. A closed system is nothing but, I would like to say, mainly it comes in case of a hematology analyzer, CLIA analyzer, some specific systems or some specific instrument where the instrument and reagent both are, synchronized or amalgamated, which means that if I am buying a product or an instrument from a company or from a party, I need to buy the reagent from that party only. That is why it is also a closed system. And this closed system concept is because of, two reasons. One, the technology and the reagent and instruments are amalgamated. Second, it will give or better accuracy or better precision of the testing when we do in closed system reagents. That is one way of classification of the instruments. Okay. It is a wide range of classification. Now let us come to the other classification. The classification is based on in which segment it is used, whether it is used in the lab diagnosis. Lab diagnosis means the central diagnosis. For example, in a hospital, the laboratory is called as a central lab. So that is called as a lab diagnosis. In India, as per the recent studies, it states that more than 100,000 laboratories are there in India, which means more than 100,000 of laboratories are there in India. So that is a lab diagnosis segment. Then comes the POCT segment, which is a point of care testing. Say, for example, a glucometer, a pulse oximeter kind of thing. No, pulse oximeter doesn't come in IVD, but it's a glucometer, of course, it will come in an IVD. But POCT reagents, which means a point of care testing, which means there is no technical knowledge required. Anybody or a common man can do the test. That is a POCT segment. Another is something called an NPT or near patient testing. Some instrument or some technologies where the instrument is taken near to the patient, the testing is done there, and data is collected. It is interpreted by the technical staff, and the result is given to the concerned. I would like to say concerned physicians. That is called as an NPT testing. Again, the instrument classification based on in which department they are used or which technology they are used. The first one is a clinical chemistry. Clinical chemistry means the instrument which is used for testing all those clinical chemistry parameters like glucose, cholesterol, liver function test, lipid function test, or kidney function test, or the pancreatic amylase test, and all those things that comes under clinical chemistry, which means these instruments are used to test the clinical chemistry parameters. Another one is called as an immunochemistry. Immunochemistry or some other classification is specific protein. Today, if you look at, there are more and more specific protein testing is coming into the IVD segment. When we started our IVD that career in 1998, the number of specific protein testing instrument in India was minimal, maybe less than hundreds. But today, more than 10,000 specific protein analyzers are there within India itself. It shows the importance of immunochemistry in the IVD. Another is immunology. Immunology is nothing but it is something like I would like to say the hormone analysis. So like CLIA platform, ELISA platform, or RIA platform. There are multiple platforms. So that we will come to the later stage. That is immunology, that is used for the hormone testing. Then comes the other segment is the hematology segment. A common segment, one of the most common segment in any laboratory is clinical chemistry and hematology. Hematology is nothing but a cell counter which is used to count the number of particles or the number of cells in the human blood, and also for hemoglobin and other hemogram estimations. Then comes the new the new segment which came to limelight after the COVID, the molecular segment. In molecular segment, there are two major technologies. One is called as a PCR technologies, which we all know, because from the beginning or for many years, and even because of COVID, we all know what is PCR, polymerase chain reaction, and all those stuffs about the PCR instruments and PCR reagents and all those stuffs. And something else is also there in this segment actually, that we also need to understand. Another is called as a LAMP, or loop mediated isothermal amplification technology, which is a technology which is there for the last 2 decades, but it is not known to the public till COVID was there. And the other instrument classification is a BGE, that is blood gas equipment, or blood gas and electrolyte equipment, which is usually used for the electrolyte analysis and blood gas based on ISC technology and potentiometer technology. And the other instrument is a coagulation system, which is used for PT, APTT, or fibrinogen, or D-dimer, this kind of coagulation assays. And the last one is a urine analysis. Urine analysis is nothing but a urinometer which is used to test the different chemical compositions in our urine. So why we have taken this many classification is that each instrument, their working principle is entirely different. So that is the reason this instrument has classified into these multiple categories. A urine analysis instrument, it works on reflectance photometry technology, whereas a coagulation instrument works on, turbodensitometric technology. A blood gas and electrolyte monitor works on potentiometry technology. A molecular test works on, again, fluorescence detection technology. It is a fluorescence is detected. Hematology works on Coulter principle and photometry. Immunology works on either CLIA or IFA or RIA or ELISA technology. Immunochemistry usually works on turbidometry, or the latest one is called as nephelometry technology, and clinical chemistry instruments works on the photometry technology. So the technologies are different in these instruments that we need to understand. So this is one of the major reasons the instruments are classified into multiple segments. And one thing we should understand, there is no single instrument who can do all those tests together in a single platform. There is no such instruments are available as on today. The reason, because different technologies or different methods are used for detecting the various parameters in our blood or other bloody fluids. Now, again, the instruments are classified into semi-automated system. That is, a manual intervention is required. Fully automated system, manual intervention is not at all required. And cartridge-based system, of course, manual intervention is required, but a minimal manual intervention is required. So these are the other three classifications. So there are multiple classifications of the instrument based on whether it is a wet chemistry or a dry chemistry, an open system or a closed system, whether it is a clinical chemistry, immunochemistry, and all those stuff, whether it is an NPT kind of thing, whether it is used in a lab diagnosis or it's a POCT instrument, and last, whether it is a semi-automated system or a fully automated system or a cartridge-based system. Why we are telling all those things is that when we look at all these instruments, the complications of these instruments are different. Some instruments is very simple. So the assuring the quality assurance, or we call it an IPQA testing, in-process quality assurance testing, is not that much very complicated. But when the complication goes, when the instrument is a different instrument or a different technology instrument or a different methodology instrument, there is always complication. And based on that complication, the IPQA testing, the raw material testing, the FG testing, all those things differs. Now let us look at the technologies which is used. In photometry is nothing but detection by means of light. It is nothing but a detection by means of light. So in photometry, what happens is that I believe the slide is visible to you. Hello?
Yes, visible.
Yeah. In photometry, what happens is that the light emitted or the light transmitted from a solution or a sample solution is detected, and that emitted data is manipulated or extrapolated based on the calibration curve. So that is why it is called as a photometry. And the photometry works on a principle called as Beers-Lambert's law. The photometry, the principle is nothing but a Beers-Lambert's law, which says that when a monochromatic light falls on a colored solution, the emergent light decreases with increase in concentration of solution and increase in thickness of the solution. So this is the basic principle of photometry. So with that principle, we use a photometric instrument to detect the different parameters available or present in the body fluids. So now the question is, the question is, if this is a simple technology, then why do we require a different instruments for, say, some similar technologies? Say, for example, reflectance photometry. Photometry also is 2-3 types. One is an absorbance photometry, another is a reflectance photometry, and another is, refractance photometry, or some we call it as a turbidometry actually. So here, a pure Beer-Lambert's law says we need an emergent ray, which means the detection is actually performed or actually done on an 180 degree to the detection cuvette. So that is why it is called as a photometry. And in photometry, we use something called as a monochromator. Monochromator, or some, it may be a filter, it may be a grating system, it may be a prism, and this prism actually will give you, or the way actually will split the light which is coming from the light source into multiple wavelengths, and there is a wavelength selector. Beyond this wavelength selector will allow only a particular wavelength of light to pass through the cuvette, so that we can get the emergent ray or the emergent light, or so that we can detect the emergent light using a PMT or using a photodiode and or PMT system. PMT is nothing but photo multiplier tube systems. So this is just very simple theory. But here also there are multiple, I would like to say variants. Variant number 1, the type of light what we are using. Variant number 2, the type of monochromator we are using. Variant number 3, the type of flow cell we are using. So these are the 3 variants, and all these 3 variants needs to be assured, quality assured, while producing the instruments. We will come to that topic later. Now, the other thing is called as a turbidometry. Turbidometry also, please understand, as I told you earlier, it is also some kind of a photometry. Only the difference is that here, instead of 180 microliter, 180 degree, we may have a detection device at, say, 90 degree or 45 degree angle. So here what happens, this kind of turbidometry instruments are used to detect the turbidity of the solution, not the, whereas photometry used mainly for a clear solution, whereas in turbidometry is used for turbid solutions or solution which contains microparticles or latex particles. And we may have multiple detector, maybe at a 180 degree, maybe at a 45 degree, maybe at 90 degree, based on the cuvette length, based on the light source use, and based on the test parameter which is performed in that particular instrument. Now, interestingly, the other one is a CLIA, or chemiluminescence immunoassay systems. Here, as the name implies, the luminescence is actually measured. The luminescence is measured. How the luminescence is measured? The luminescence is measured when, say, for example, we have a target antigen. So there will be a capture antibody available in the reagent. So the capture antibody will attach to the target antigen. Then there will be something called as a detection antibody there. And this detection antibody and the target antigen, the capture antibody, they will form an antigen antibody complex. Into that antigen antibody complex, we will add a luminous substance. We call this a substrate. When this substrate is reduced using a reducing agent, when the substrate is reduced, a luminous is produced, and the luminous is measured to detect the amount of antigen or antibody, whatever is present in the human fluid, the body fluid. Why this technology is different is that the photometry or turbidometry systems, the reactions are 3 types. We call it as an endpoint reaction, a fixed time or a fixed time reaction or kinetic reaction. In CLIA-based system, there are only two technologies. One is we call it as a flash technology, and another is a glow technology. Flash is nothing but when you add a reducing substance or the chemical to create the luminol. What happens? The luminous will go like this as a curve. It will reach a paramount. Then it will be there for some time. That is why it is called as a glow. It will act like a glow. And something else is there as a flash. Flash means when you add the substrate and then you add the reducing reagent, there will be a flash. That's all. So here, because here the technology is different and the technology, the flash itself is different, two different types of PMT tubes are used to capture these kind of reactions. In photometry or in turbidometry, most of the parameters are measured in milligram percentage and gram percentage and in microgram percentage, whereas in CLIA platform, the particles or the parameters are measured or the proteins are measured at micromole or nanomole kind of measurement. So because of this, the PMT also will be different, the detection technology also will be different, and the complexity also will be different. Another one is called as an FIA. We talked about CLIA, which is chemiluminescence. FIA is fluorescence immunoassay. Here also, what happens? The process is somewhat similar. So instead of forming a chemiluminescence, it will glow using a fluorescence immunoassay or a fluorescence, and this fluorescence is detected and the parameters present in the blood is quantified based on the fluorescence. Now, this is very, very important thing. So far, we have discussed about different terminologies like, photometry, turbidimetry, immunofluorescence assay. Then I would like to say that chemiluminescence immunoassay. These are all based on the photometry or optics. We are detecting either the emergent light or the refracted light or the glow or a flash. But here, a flow cytometer or a Coulter counter, actually what it do is that it is a particle counting technology. So the particle counting technology is very, very, very simple. It is a simple technology. There is two different troughs or two different flow cells. We call it as a flow cell. And in the flow cell, there will be a fluid. And there is a small orifice is present in the inside flow cells. And there are two electrodes. When any particle, for example, particle cell counter is used in hematology. So blood particles like platelet, RBC, WBC, these are the three things: RBC, WBC, and platelets. Okay, whenever there is an RBC or WBC or platelet, whenever it passes through this orifice, what happens is that there will be a pulse generated because of the change in the resistivity. When it passes through the aperture, we call it as an aperture. So the pulse generated is directly proportional. The pulse generated is directly proportional to the size of the cell, number one. And the number of pulse generated is equal to the number of cells passed. So that is why it is called as a particle counter or a cell counter. Okay. So here the technology used is entirely different. We call it as a Coulter counting. So this technology is used in all hematology analysis across the globe for counting the cells. Now, come back to the flow cytometer. Now we are counting number of cells in the body, in the body fluid. Now we need to identify what are all those cells present in the body fluid. For that, we use the flow cytometry. In flow cytometer, what happens is that when the cells are allowed to pass through a straight line using a hydrodynamic focusing, the particular technology is called as a hydrodynamic focusing. When the cell passes through this flow cell through a using a hydrodynamic focusing, a particular wavelength of light is hit on this these cells, and the emergent light or the scattered light is detected at multiple angles. We call it as a forward scatter, we call it a slide scatter, wild angle scatter. These are the three, or fluorescent scatter or FSC. That a scattered light is captured. And from the scattered light, from the captured scattered light, there is an algorithm. And with that algorithm, we will detect, or we will identify which type of cell it is. So this is called as a flow cytometry. So in an instrument, the Coulter counting is for the number of cells or the number of particles, whereas in flow cytometer is to identify what is the cells. Because in the human blood, RBC is one particular cell, platelet or thrombocyte is another cell, WBC or leukocytes, there are different cell lineage like neutrophils, eosinophils, basophils, leukocytes, monocytes, and also others like other cells like band forms or myeloid series, or the lymphoid series, which are abnormal cells or cancerous cells, etc. So that is the reason a flow cytometry is used to detect the cellular type. So these are two different technologies. Now we have heard about different technologies which is used in the instruments, right? Now, why we have said these different technologies used is that to assure the quality of the instrument, we need to understand first what is the technology which is used. And based on the technology which is used in the instrument, the quality control, the IPQA, the FG, the RM material inspection, everything is different. Please understand and please note this as a point. If I am going to validate a clinical chemistry instrument, there is an X number of ways. And when I go to validate a hematology instrument, there is an X number of Y number of ways. When I'm going to validate a CLIA instrument, there is, say, Z number of ways, because the methodology is different, so that the calibration or checking is different in different instrument based on the technologies used in the instrument. Yeah. Now, the term quality refers to a degrees to which a set of inherent characteristics fulfill the requirement. This is not my words. It is taken from ISO Vim, Vim Gate. So when I say quality product, which means that it is having some inherent characteristics, correct? So how do we assure the quality is the second question. Now, if I'm a manufacturer, for example, I'm a manufacturer. It is not an example. Agape, we are a manufacturer. We are manufacturing automated instruments. We are manufacturing cell counters. We are manufacturing coagulation system. We are manufacturing urine analysis system. Then we are manufacturing hematology analyzers. We are manufacturing specific protein system, CLIA. So far, we are not manufacturing. We are manufacturing clinical chemistry system. So how do we assure? Okay, whatever we manufacture in our facility, it is at par, or the purpose is, it is met. So let us go through that process. Now, the question to entire team, I think, I request a participation from the team. What are we looking on an IVD test? I request, the team to give your answers, please. What are we looking for an IVD test? Anybody can answer. Am I audible? Hello?
You're audible, and anybody can unmute and talk.
Yeah, please unmute and talk. What are we looking for an IVD test? Because without knowing what is our goal, how can we assure the quality, my dear? So what are we looking for an IVD test as per you? Now I will give you a different questions. When the number of when the questions we started asking, I think some of the participants left, so no problem. But still, I will ask you questions in a different way. So you are going to a laboratory. See, any of you are going to a lab. Now you are doing a test. So what you expect from the test?
Accurate results, sir.
Exactly. This is the answer actually I am looking for. We require an accurate result. Correct? Result accuracy. Anybody else?
Without impurities to get results.
Without?
You mean interference?
Without interference. You're right. Absolutely right. Yes.
Repeatability?
That is precision or repeatability. Yes. Any more wild guess?
We should get a reliable result because we should not get any false results or anything like that.
Exactly.
Because,
exactly.
When we are going for COVID testing and everything, then
Yes.
if it is negative, positive, then it is a problem. So we should get a reliable result. We should not get any false results, etc., etc.
Yeah, true. Actually, when you look at, any IVD testing, actually we are looking into three things. One is an accurate result. The result should be accurate, which means, okay, when I do my COVID testing in next lab, if it is coming positive, it should be a real positive. It should not be a false positive. And it should not be a false negative, because false negative is dangerous than false positive. I think, you people can, you are the people who can give more comment on this. I'm not the person to comment upon that. Second thing is it should be a precise. Our result should be a precise result, which means repeatability. Then third is the resolution. Why I said resolution? Because in hematology instruments, resolution plays when it comes something called as there is something called as the scatter plot. When the scatter plot comes, then resolution plays an important role. So in an IVD results, we require accuracy, we require precision, and we require resolution. So that is acceptable to all. Now, when we manufacture a product, so what is our quality criteria? The instrument should give an accurate result. The instrument should do a precise result. And the instrument should have a resolution. Correct me if I'm wrong. So when we do a quality assurance, when we make a QA protocol, quality assurance protocol, our primary aim is to make sure that, yes, the instrument is giving an accurate results. The instrument is giving a precise results. The instrument is giving a highly resolution results. So let us see how we are going to validate an instrument. We call it as a validation, instrument validation. There are certain criteria for do the instrument validation. Which means whenever I am manufacturing an instrument, the instrument is validated at factory or by the QA or by the QC team or the FGQC is finished good QC team to make sure that the instrument meets the intended use or the criteria. Now, how we are going to do on what are the quality control steps? What we are going to do in this kind of things. So you know the difference between QA and QC. QA is a proactive approach at the process level and QC at the product level. So we need to do both at the process level and the Q and the FG level. So now what at the process level, how do we do it? We have certain multiple tools. We call it as a quality tools. We have control chart, scatter plot, check sheet, Pareto chart, flow chart, cause and effect diagram. CED will come especially in an RCA or on a root cause analysis. Then control charts and histogram and a scatter plot. All this comes as in FGQC, whereas the Pareto chart comes in QA, especially in an IQA, and check sheet comes in IQA. A flow chart is also comes in QA, because a flow chart actually define, okay, these are the criteria, or these are the SOPs to be followed while manufacturing an instrument. So that is a flow chart. And check sheet, individual processes needs to be checked. Now we have guidelines. There are guidelines which is available in the industry. We call it a CLSI guideline. CLSI is nothing but Clinical and Laboratory Standards Institute. It is a US organization. Earlier it was known as NCCLS, National Committee for Clinical and Laboratory Standards. So earlier it was NCCLS, now it is on CLSI. So there are guidelines for each and every such kind of processes. So for example, hematology. If I am manufacturing hematology, if I am giving a hematology instrument to the market, before giving that product to the market, I need to make sure that the instrument is working as per the specification, and the specification is H26A2. That is the specification. Whenever I am manufacturing a product, I am giving this product to the outside world. If I want to make sure that the instrument is working with the reference standard, I need to do a method comparison studies, which is EP0982. Now, for example, if I am using a CLIA instrument where, say for example, something called as the best example is PSA, prostate specific antigen. If I want to report a PSA, I need to make sure that my LOD, my LOB or limit of blank, my LOD or a limit of detection, and my LOQ or limit of quantification is actually satisfying the criteria given by the international organizations. Now, if I want to make sure that yes, my instrument is giving a precise and accurate results, I need to follow the guideline EP05A3. If I'm telling that yes, my instrument is giving a linearity of, say, glucose from 45 to 600, to say that my instrument will give 600, I need to do a study as per EP06A. So there are guidelines available in the industry for doing this kind of test. Now, we use some common statistics like mean, standard deviation, coefficient of variation, and regression stat. These are the commonly used statistics for performing this validation protocol. So how do we validate? First one is the physical validation. Physical validation is nothing but the appearance. Say, for example, if I am making a product, first I will make sure that by appearance, if there is any problem, then we will do the performance validation. The performance validation, there are around 6 different parameters. One is the method comparison, another is the sensitivity, third is the linearity, fourth is carryover, accuracy studies, and precision studies. So all those studies we will do whenever we make any automated instruments. Now, what is the manufacturing process flow? The manufacturing process flow will be raw material to IPQA to FG. So from raw material to IPQA, there are some processes. From IPQA to FG, there are some processes. Now let us understand what are the processes in a raw material testing. When in a raw material, we call it as an IPQA or not IPQA, in inward quality control or IQC, where we check each and every components using specific jigs. We call it as jigs because in case of an instruments, the raw material is nothing but electronic components. So these electronic components needs to be tested using specific electronic jigs. So we use specific electronic jigs, which is customized for each and every instruments. So for example, if I'm getting a board, this board needs to be tested with using an electronic jig. If I am getting an IC, the IC needs to be tested using an electronic jig. If it is a PCB or printed circuit board, the PCB needs to be tested using an electronic jig. Now, another is, this is very, very important, the factory calibration. How do we do the factory calibration is very important for, assuring quality of the instruments, IVD instruments. And let us see what are all the things we calibrate. Number one is the photometric calibration. For photometric calibration, there are international standards available for calibrating. There are colored solutions, like say, for example, perchloric acid or chromate solutions, or there are certain colored solutions available in the international standards as an international standard, which is used to measure or which is used to calibrate the photometry. So photometric needs to be calibrated using these kind of standards. Now, the second is calibration of pump or calibration of the diluter. How we make sure that the calibration is or the pump is working perfect? We need to use the calibration. Now, a simple example I'll tell you. There is something called as infusion pumps in the medical industry. I think you people know that more about that infusion pumps. So what is the infusion pumps? The infusion pumps will inject a particular air or a definite amount of fluids into the body. Correct? So infusion pumps, their major or the heart of the infusion pump is nothing but a peristaltic pump, which is shown here. Actually, this peristaltic pump is actually controlling the flow rate to the body. Similarly, in automated instruments also, there may be peristaltic pumps or bellows pumps or some way vacuum pumps or something, ceramic pumps. There are multiple pumps available. Different types of pumps are available for instruments. So these pumps needs to be calibrated properly so that the proper or the perfect amount of reagents or fluids are aspirated or dispensed while doing the testing. So the number one thing is calibration of pump and diluter. So for example, if I am going to say, for example, let us take an example of assembling a chemistry analyzer. In a chemistry analyzer, there are three major components. Number one is the PCB or printed circuit boards. The PCB first checked, individual PCB is checked whether the PCB is working or not. And please understand, something else is also there. We call it as in our terms called as an it is called as firmware or the software which actually runs this instrument. That software is loaded in the PCB, if the software is working perfectly or not. Second thing in any such instrument is that the pump, whether the pump is working perfectly or not, the pump calibration. Otherwise, what will happen? Can you just imagine a case? We program to pump, 10 ml in 1 hour. Okay. But if the pump is not properly calibrated, instead of, pumping a product in 1 hour, it may pump a product maybe in half an hour. So which means we need a calibration of a pump or a diluter. So there are certain standards is there for pump calibrations. Once the pump is calibrated properly, the calibrator or the person or the technical staff who done the calibration properly, he will write pump calibration is done, and he will put some value which we called as the default value. So we will call this a default value. So default value, say for example 0.709 offset. We call it as in our terminology as an offset value. Now, calibration of a flow cytometer. See, in flow cytometer or in Coulter, what we are actually measuring is nothing but the particles or the different sizes, correct? So here we use a particles itself for calibrating this thing. There are international standard particles available of multiple micrometer length, say maybe 6.82, 10.2, like that. In multiple micrometer length, micrometer radius, particles are available, or standards are available, and these standards are used to calibrate the flow cytometer. So, for example, if I am using a particle with 10 microliter radius, when this particle goes through this flow cell, it should give a signal to a 10 microliter chamber, which means that the flow cytometer is calibrated properly. See, and please understand one thing, in case of instruments, most of the things what we do is, engineering calibration. So this calibration we have done, we need to do. So otherwise, if the calibration is not done properly, the flow cytometer will fail to give the signal at the particular wavelength. Now, IPQA or in-process quality assurance, like the checklist what is shown here, each and every process, the concerned IPQA person will make sure that the offset or the points are well noted, it is within the acceptable limits, or else they will reject it. Because we need to make sure that all those values are within the acceptable limits, or else they will reject that particular product at the IPQA level itself. Now, when it comes to the FGQC, here comes the maximum testing using reagents. Till the IPQC, from the raw material to the work in process material, it is IPQA job. When it comes to the FGQC, we do all this kind of testing. So we do a method comparison stat, we do a sensitivity stat, we do a linearity stat, we do carryover studies, we do accuracy studies, we do precision studies, and we also need to do a GUI validation, which is called as graphical user interface validation. So these are the validations we will do at the FGQC level or final QC level. Now let us see how do we do accuracy. Okay, now how can I claim that, okay, my instrument is giving accuracy? There are two methods available. One is the using sample, another is using control. There are control materials available for accuracy. We call it as a linearity control or accuracy controls. And each accuracy control is having a control value along with the range and for each parameter. Say, for example, if I made an instrument, say for a glucometer, I want to measure the glucometer is working perfect or not. I'm using a control and the control value is 200 and the range is given 200 plus or minus 20. I tested this control in my instrument. It given 206, which means my instrument is proper, which means my instrument is okay, and I will release this instrument with the release note. We will come to that later. Then using a sample, if some reagents or some product or some parameter, a quality assuring quality using control material won't be a good thing, because the availability of control will be an issue. In that case, we use sample metrics. We collect sample, we do the sample test in our instrument, which is called as a test instrument, and we do sample in the reference instrument, which is called as the reference instrument or the reference method, and we do in statistics. And if in the statistics, if the correlation coefficient is greater than 0.95, then which means that our instrument is giving an accurate results. So this is two type of methods what we use, either by using a sample or using a control. Now, precision. For precision, what we use is a CLSI EP5 guideline. So we need to check within a day precision and a between day precision. Why this precision is required for an instrument is that if the instrument is giving a precise report, it means that the instrument hardware, including the photometry, including the tubings, including the GUI, all those things are working perfectly. That is the importance of precision. So for that, there are multiple methods available. Usually we do a 20-day studies where the control material or some pooled sample material, we will run for 20 days. We will take the data and we will interpolate. We will take the standard deviation, CV percentage, and if the CV percentage is less than the acceptable limit, there is a acceptable limit given by the international chapters. We call it as total allowable error, or TE small a, total allowable error. If the total allowable error of the parameter, whatever we are testing, is within the acceptable limit, then we can say that, yes, our instrument is precise. Now, this is in a typical example of a method comparison. One side you can see the bias plot, which means the replicates, and other side you can see the correlation coefficient. These two plots actually tells that, yes, the machine, whatever we have manufactured or we have produced, is passing the QC as per the QA standards. Now, the second is the linearity. For linearity, we take the rate of reaction, and we see if the rate of reaction is proper. If the rate of reaction is proper, this indicates one important thing, that our photometric calibration is better, our temperature calibration is proper, our flow cell calibration is proper. All those things are actually detected by means of linearity studies. Now let us consider carryover. So carryover is nothing but, say, I'm doing a test, correct? So all the instruments, I'm doing multiple tests. So some instruments, there will be a semi-automated, we will use a test tube, and each time we need to feed the test tube, feed the reagent from the test tube or the reaction mixture from the test tube, and the reaction mixture or the chemicals is aspirated by the instrument, and after aspiration, it is detected and it is expelled back to the waste. But what if the reagent is not expelled properly by the instrument? What happens? The chances is that, carryover will happen, which means the previous reagent will interfere with the new reagent, creating carryover. So we need to make sure that whatever instrument we are producing, especially this is applicable for, instrument which uses, wet chemistry reagents, where aspiration, dispersion, and all those things are there. So in that case, we need to make sure that there is no carryover. So for that, we will do a carryover study. And as per the standards, a carryover less than 2% is considered to be acceptable. So if the carryover after our experiment is less than 2%, which means that, yes, our instrument is working perfect. There is no carryover, which means that, there is no residual error happening inside the system, whether it is a fully automated system or a semi-automated system. There are two types of carryover. One is called as a sample carryover, another is called as a reagent carryover. There are two types of carryover, and when it comes to certain instruments, there is another type of carryover called as a cuvette carryover. That is cuvette carryover study, then the reagent carryover study, and the sample carryover study. In reagent carryover study, one is called as a recipient and another is called as a donor. Donor is the reagent which donates carryover to the recipient, and recipient is the reagent which gets interference because of the donor chemicals. This is very, very important in fully automated systems. This carryover studies very, very important, because if this carryover study is not properly performed, many a times it can give erratic results. So this is very important. Another one example is something we call as an OSS, or optimized smart sampling. This is in some instrument we call it as an OSS sampling, where, for example, sample A is creating a problem to sample B. So what the instrument will do, it will take sample B first, then sample A, then sample C. So which means that, the carryover is minimized. So this is a carryover function. So this study is it's a big study. Actually, it is not a simple study. We need to find out which all reagents are creating carryover, and which are combinations it is creating carryover. And it is a huge study we need to do whenever we design usually an instrument. Usually most of the studies, these carryover studies, linearity studies, will be performed at the time of design level. When it comes to the mass production level, majority of the thing, it is an accuracy studies, accuracy and precision studies. Carryover studies or other linearity studies is not required, since at the time of designing the instrument, at the design phase itself, or beta 1 or beta 2 phase, all these kind of studies are performed, and and the data are taken, all those corrective actions are taken, preventive actions are taken before passing the same to the production line. Now, another is the detection limits. Why we have taken the detection limit is that, this is very important factor. This is very important factor is that, see, if our idea is our result should be accurate, our result should be precise, and it should have a good resolution, correct? So these are the three major goals for our instrument. Now, let us take a case of a prostatectomy. So somebody removed the prostate from a patient, which means there should not be PSA available or prostate-specific antigen available in the serum, in the sample, right? So if the instrument is not capable of detecting very low amount of PSA, then it will be always a problem to the practicing clinicians. So that is why whenever this kind of parameters are tested in instrument, we usually do an LOB, LOD, and LOQ studies. If any parameter, if it is less than LOB, it should not be, the result should not be given to the patient. Instead of that, you should write not detected. If it is LOD or limit of detection, you can only say detected. But if it is more than LOD and up to LOQ, then only you can actually quantify that, yes, my instrument, my instrument is giving a value 0.25, which is accurate and precise. So these kind of studies are also required to assure the quality of results, whatever we are delivering when we manufacture an instrument. So please understand, most of the studies are performed at the design level to make sure that the instrument is performing well. Now, another one is called as a GUI validation. The GUI validation, in the GUI validation, it is not only the GUI validation, there is something called as an LIS connectivity. There is something called as an remote connectivity. All those things are, are checked at the time of, GUI testing. So say, for example, when I touch something, the command whatever is given in the panel, and the command whatever is going to the system should be same, correct? So in order to make sure that yes, the perfect command is going to the system, we do a GUI validation. Again, I'm telling you, the complete GUI validation will be performed at the design level, and when it comes to the production level, only a routine GUI validation using, multiple criteria is only performed. Now, there is something, in an instrument, we need to make sure that the instrument is giving proper result, right? So this to make sure that instrument is giving a proper results, there we provide a user manual. Why? Because if there is any query by the customer can refer the user manual and he can find out the remedy. The thing why we give a bigger manual, when it comes to the reagent or another simple thing, it is an insert, an IF or instruction for use. It is a small insert, maybe a 4-page or 5-page. But when it comes to the instrument, we give a complete user manual depicting all the flowcharts, all the menus, what are there in the menus, what are the do's and what are the don'ts, so that the customer or the client or the patient or the physician, whoever is actually using that instrument, can understand what went wrong and how we can arrest it. Now, the service manual. Of course, the service manual is a part of the service team, but still we give a service manual to the client to make sure that any issues like for small servicing can be addressed properly. And now comes something called as an IQ, OQ, PQ document. This document is required by, or by these third-party auditing agencies like NABL, or Quality Council of India, ISO, all those things, or Intertech or RCPA, whoever is the third party audit, they require this document. IQ, OQ, PQ document is nothing but a document which says that, yes, the instrument is installed properly, that is IQ, installation qualification. OQ is the operational qualification. Yes, the instrument operation is perfect, and PQ is the performance qualification, which means the instrument is performing properly. Please understand, or whatever we have discussed earlier, all those things are performed at the factory level or the company level or the manufacturer level. Whereas IQ, OQ, PQ document, this document is made when the instrument is installed at the customer or laboratory or hospital, wherever it is. So this actually makes sure that, yes, the results, whatever is giving by the instrument, the quality is assured. That is why we use a PQ or performance qualification. Then, of course, we will give a maintenance checklist, a user maintenance checklist, a service maintenance checklist, and a preventive maintenance checklist, along with the manual, to make sure that the results given by the instrument is having a better quality. So now, earlier I said, whatever test we do, we give actually a release note. So this is a typical release note where we call it as a certificate of analysis or release note, some different criteria or factory release note. So here we will give all those details. What are the major things? Because, I just given it as a sample, because each instrument, say for example, typical example of CLIA. In CLIA, we will write RLU, or that reflected light units. In IFA, we write FIU, or fluorescence immuno unit. When it comes to photometry, we will write it in absorbance, or ABS. All those three are different. So the values or the default values also will be different. So based on the technology, based on the values, we will mention whether the instrument is gone through the standard checking, and whether the instrument is is passing the standard, whether it passed the FG control, so that can be used for the in the laboratory. That is, we call it as a release note. And all those release notes, there are two things. Sometimes this release notes will be along with the instruments in a pouch, or sometimes the release note will be with the manufacturer in their files. So there are two scenarios. Now with this, I think we will conclude the session. So any questions means please you can ask.
People can unmute and they can talk.
Yeah.
Nobody wants to have any questions? Dilip, you can just. Nasser?
Sir, I think there is no question in chat box.
Yeah, no question. Not seen in the chat box.
Okay.
Either the things are very tough or they're not clear with the topic. I think many are not familiar with the topic, isn't it?
Yeah, true. I agree to that, because here in instruments, lot of engineering stuff is also there. Because electronics.
It is a good topic, and it was very informative with respect to the quality assurance.
Yeah.
Because the field is different, so nobody is asking question.
But
Okay. What is the frequency of this performance calibration for IVD equipments?
See, usually the calibration, say for most of the equipment, once in a year is a calibration is required. But in some instruments, like this hematology instruments and all, you may require a calibration once in 6 months, but all these calibrations required if the control is going out of range. Otherwise, usually calibration is not required.
Okay.
Because it is
So-
instrument specific. It is instrument specific.
Okay.
Sometimes, it is region specific also. Say, for example, if you buy a glucometer, every glucometer strip, there is a calibration card.
Yeah.
Like that. So it is instrument specific and technology specific.
So they have to do frequent verification for the performance. Only.
Pardon?
They have to do verification of the performance.
Yes.
Using controls.
See, yeah, with control. See, in say, I'll give you a typical example of glucometer. Glucometer will have a control strip, correct? No?
Yeah.
So that is why the control strip is there. So there in glucometer, it is based on biosensor technology. Okay. So since it is a biosensor technology, the control chip is also biosensor. So when we insert the biosensor, actually it is checking whether the internal algorithm is correct or not. That's all.
Okay.
Whereas when it comes to some hematology instruments, we have controls available. 3 levels of controls: L1, L2, and L3. Level 1 or the very low, level 2 is normal, and level 3 is very high. So all the 3 controls we will do, and if the control falls within the area, then within the range, which means that instrument is working fine, like that.
Okay.
In different equipments or different categories, the normal value or the range of normal value is found different sometimes. What is the basis of that?
See, the reason is that earlier I told you, sir. So here, even different instrument is having some different technologies. Say, I will give you a typical example. Glucose is having 2 different technologies. One is a GOD-POD method, another is a hexokinase method. In both the methods, if you do the same patient, same sample, same instrument, there will be a plus or minus 10 variation. Say maybe 5% variation is there, and that is an acceptable variation actually. That is there. Then another one is thing is that instrument to instrument. Say for example, in hormone, there are 2 different technologies. One is a CLIA, chemiluminescence immunoassay. Another is eCLIA, electrochemiluminescence immunoassay. When you do a test in eCLIA and when you do a test in CLIA and when you do the same test in IFA, the range will be different and the reference range will be different. Their units will be different, but the interpretation will be same. Like that.
So every time you have to refer that particular range.
Yes. This is applicable especially for CLIA systems, because for CLIA there is no international standards like chemistry. Chemistry, there is a standard, NIST, that SRM 909B, I don't know what is the latest lot number. SRM is there, SRM, CRM is there. But for CLIA, unfortunately, there is no such SRM, CRM, which can be commonly used. That is one reason.
Can you explain what is the NGSP we call in terms of the HbA1c test?
See,
What is that?
NGSP means, National Glycohemoglobin Standardization Program. It is a standardization program by the Diabetic Association, International Diabetic Association, where they are making sure that the HbA1c value, whatever we are giving, is in accordance with the international standards. It is a standardization program. Otherwise, when you do an HbA1c in multiple technologies, I'll just give you a typical example. Purposefully, I have not taken HPLC. HPLC is also comes under IVD, but that subject is very huge. HPLC, that high performance liquid chromatography, that we usually use it for that is for protein, for hemoglobin, and especially for HbA1c. The thing is that if you do an HbA1c using an HPLC, and when you do an HbA1c using a column chromatography, and when you do an HPLC using a latex enhanced immunoturbidometry, and when you do a HbA1c using a cartridge-based method, all the HbA1c values, there will be plus or minus sometimes 1% deviation. And to minimize this deviation, all these instruments are actually calibrated against an NGSP. So that is a traceability matrix actually. Because of NGSP, we have a traceability matrix.
Every parameter there is a NGSP or traceability material available.
Yeah, see, most of the clinical chemistry parameters, there is a traceability matrix available. Most of the clinical chemistry parameters, there is a traceability matrix available. But for hemoglobin also, I believe that traceability matrix is available. But when it comes to CLIA, there is no single traceability matrix available as of now.
There is a question in the chat box.
Yeah.
I have heard that LOD and LOQ is not always the same for instruments. Where does it differ? Is LOQ more concerned in analyzing impurities?
Okay. Thank you, Muhammad Danish. I will just show you the slide. One second. Let us go back the slide. Yeah. This LOB, LOD, and LOQ is nothing but the detection limits. Okay. It is called as a detection limits. Limit of blank means, say, for example, I am giving a blank water or blank sample. When I do give a blank sample, what is the value I am getting for a blank sample? That is LOB, limit of blank. So which means that, say, for example, if my glucose is always to, say, if I give a water to a glucometer, it will give 2, which means that the limit of blank is 2. Clear? Now, LOD or limit of detection. Limit of detection means, okay, it is detected, but precision is not there. So, for example, when I give a value, then a sample with a value, then it will give, say, 10 as a glucose. It is detected, but it is not precise. That is why called as a limit of detection. So, limit of quantification means if I give my value, my sample, it will give 50, 51, 52, 51, 52. Precision is there, accuracy is there. That is called as LOQ. Sometimes both LOD and LOQ will be same. Sometimes LOD and LOQ will be same, but LOB will be always different because LOB is used on blank sample, whereas LOD and LOQ is tested using samples which is having analyte in it.
Then what
I hope that it is clear.
Upper limit? Upper limit, then what happens? Is there an upper limit?
Upper limit is something called as linearity. It is called as linearity, or in terminology, it is called as AMR, analytical measurement range. AMR or analytical measurement range. Say, for example, instrument to instrument AMR differs. Say, for example, a semi-automated system, because of its hardware, because of this software, it may be having a sensitivity. Okay. If I use that semi-automated system, my sensitivity will be X. If I use a fully automated system, my sensitivity may be 10X or 3X, not 10X, 3X. So for the same parameter, linearity in a semi-automated system will be 500. Sorry. Or for the same parameter, the linearity in a fully automated system will be 1000, something like that.
Now, what I have noticed, that pharmacy students basically are not conversant with these systems. I think in the curriculum also, Sanjay Mohan, we will have to have something like this. I used to tell our people. Krishna Kumar.
Actually, the thing is that. See, even last week I was in Nepal. Giving the lab IVD team on quality assurance only. The thing is that earlier there was something only quality assurance, QA and all. Now today everything is changed to TQM or total quality management. In total quality management, there are multiple systems we need to understand. And like say for example, when you come to an HPLC is a study material for the pharma students. Correct me if I'm wrong. But how can you identify that whatever the HPLC is giving perfectly or not? So we need to have some kind of, this kind of, deeper studies onto quality assurance. This is my simple request.
Anybody from academics to talk on this, Krishna Kumar, Dr. Krishna Kumar? Because I generally see that normally people are not coming to these aspects, even though the quality control, when you have quality control in the pharma also, it is a bit different than what you normally see here, isn't it, Dilip and Nasser? In your laboratory, in our laboratory also, almost the same things are being applied?
Yeah, sir. But we are these IVDs are not we are controlling.
But
Because it
all
is not coming.
all these systems
Yeah,
are
parameters are same. Precision and also this assurance levels. These are same. Calibration and performance.
And any other courses given by our PG course available on these things for pharma? No, no. Any you know?
No, sir.
No. So no questions are there, Dilip? And students? Silent.
If there is no more questions, we can next move on to the next level.
Yeah, as a moderator, I mean, Abdul Nasarji, anything to comment and then?
Yes, sir. It is a very informative session, and I think it is our second section about this in vitro diagnostics. Earlier, it was on reagents, because in vitro diagnosis will plays an important role in the healthcare system, and so its quality assurance also plays a vital role. Our speaker Sanjay Mohan given a very informative talk about this, and his presentation also was very nice. Thank you, Sanjay Mohan, sir. And that's also. Now we can move to next.
Yeah. I think we can have an offline session also in between for one day. I was talking with our DC also for our analyst in the government service as well as private companies. We will have Sanjay will have a one-day program.
Actually, sir, what my suggestion is that we should have some kind of a program on an SPC, statistical process control. Because say now many sites, there are SPCs available in the curriculum, like Shiva charts or LJ charts or whatever it is. We are actually plotting. But what we need to now look into that, after plotting the chart, what we are actually doing. Are we just analyzing the chart, or whether we are doing a kappa, that kind of a thing, we need to do more on an SPC.
Yes, we'll think over, I think, next time.
Yeah.
So over to the team.
Thank you so much, sir. That was really an interesting session, very informative. Let me also take this opportunity to thank Sri Abdul Nasser, sir, who is currently working as Head of Instrumentation and Quality Manager. Now, I would like to call upon Bharat Narayanan, Final Year B.Pharm, Prime College of Pharmacy, Joint Secretary, Students Forum, and Amada S., Final Year B.Pharm, Prime College of Pharmacy, Director, Professional Relations, IPGA Kerala, State Branch, to say a few words about the session.
Bharat, not hearing. Some sound issues there. You can use your mic straight directly. Yeah. Yes.
Good evening to one and all. We found this seminar incredibly valuable. We have learned a lot of detailed information about IVD and its importance. In the current scenario and over the last several years, as we have been through the most challenging situations of COVID-19, the RT-PCR test kits helped us diagnose the disease earlier and a lot quicker, which shows the importance and need of IVDs. We look forward to using what we learned in this session. Thank you, sir, for providing us with your valuable views and your valuable time. Thank you, IPGA and KPGA, for having me to the part of this session. Thank you, sir, and thank you.
Now.
Thank you, Bharat. Now, we are at the official end of our program. So, to propose vote of thanks, I call upon Shaymol, Pioneer B.Pharm, Prime College of Pharmacy, Vice President, Students Forum, IPGA Kerala State Branch.
Good evening, all. It gives me an immense pleasure to deliver the vote of thanks. I would like to thank Mr. Abdul Nasser K, who was our moderator today, and I would like to thank Mr. Sanjay Mohan KV for the review. And I would like to thank all the officers and member of IPGA and KPGA. And finally, I thank all the students and others who attended the webinar. Thank you all.
Thank you, Shaymol. You don't need to be a brain surgeon to become a valuable person. You become valuable because of the knowledge that you have. And that doesn't mean you won't fail sometimes. The important thing is keep on trying. And I would like to use this chance to thank everyone behind this webinar on behalf of IPGA and associated with KPGA. Thank you all. And we end this section with National Anthem. Signing off, I am Sneha with Nikirti Mohandas. Thank you all.
Sneha, you can sing, otherwise all can sing. Our people can sing in case it's having difficulty in the sound.
Okay, sir.
Okay, sir.
Yeah, please. Start, Bharat. You can start.
Yes, sir.
1, 2, 3, start. Jana Mana Adhinayaka Jayagi Bharata Bhagya Vidhata Punjab Sindhu Gujarat Maratha
India Himalaya
Yamuna Ganga
Uchala Jalati Taranka Tava Shubha Name Jage Tava Shubha Shisha Mage Gahe Tava Jaya Gata Janagana Mangala Dayak Jaya Bharata Bhagya Vidhata Jaya He Jaya He Jaya He Jaya 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 He.
So thank you, everybody.
Thank you, sir.
Thank you so much.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you, sir.